Hello and welcome. Today we will explore culture-independent methods for studying microbial communities. In previous part, we explored culture-dependent methods where microorganisms are grown in laboratory to study them. However, this approach has some major limitations as discussed previously, such as most microbes cannot be grown under standard lab conditions. In fact, over 99% of the microorganisms remain unculturable. So the question is, how do we study the vast majority of microbes that we cannot grow? Or how do scientists study this hidden majority? This leads us to culture-independent methods, which allows direct analysis of microbial communities without the need for cultivation. The concept is simple but powerful. Instead of growing microbes, scientists analyze their genetic materials and other biomolecules directly from environmental samples like soil, water, human guts, etc. These biomolecules include DNA, RNA, proteins, and metabolites. For example, imagine analyzing a drop of pond water. Without growing anything, we can identify all microbes and even understand what they are doing. This approach allows us to study entire microbial communities, including those that cannot be cultured. Culture-independent methods can be divided into several major categories based on what they analyze. These include microscopy-based methods, DNA-based methods, and RNA, protein, and metabolite-based methods. Each approach provides us a different layer of information about microbial communities. Let's start with microscopy-based methods. Microscopy-based methods allows us to directly visualize microbes in their natural environment. One important technique is FISH or fluorescent in situ hybridization. In this method, fluorescent probes bind to specific DNA sequences inside microbial cell. Under a fluorescence microscope, labeled microbes become visible. This allows scientists not only to identify microbes but also to see where they are located within a sample. So unlike sequencing alone, this method provides special information. For example, in a dental plaque sample, FISH can show exactly where different bacteria are located, some forming layers and other clustering together. So this method tells us not just who is there but also where they are. Now DNA-based methods. The most widely used approaches are DNA-based methods. These involve extracting DNA directly from a sample and analyzing it using sequencing technologies. These methods provide detailed insights into microbial identity and potential function. Let's look at the key techniques of DNA-based methods. One of the most common DNA-based approaches is 16S rRNA gene sequencing, most often performed as 16S rRNA Amplicon sequencing. Here, scientists target a specific gene, the 16S rRNA gene, which is present in all bacteria. By sequencing this gene from environmental DNA, scientists can identify and classify bacteria present in a sample. This gene contains both conserved and variable regions. The variable regions act like a fingerprint, allowing us to distinguish different bacterial species. For example, in a human gut sample, this method can identify hundreds of bacterial species at once. So this method helps answer a fundamental question, that is, who is present in microbial community. Another powerful approach is shotgun metagenomics. To go beyond identification, scientists use shotgun metagenomics. Instead of targeting a single gene, this method sequences all DNA in a sample. This provides both taxonomic, that is, which microbes are present, and functional information, that is, what gene they carry. For example, in soil samples, shotgun metagenomics can reveal genes involved in nitrogen fixation or antibiotic production. This allows us to understand not just which microbes are present, but also what genes they carry and the functions they may perform. So it answers two questions. One is who is there and what they can do. But having genes doesn't always mean they are active. To understand real-time microbial activity, scientists use metatranscriptomics. While shotgun metagenomics shows potential functions, metatranscriptomics reveals activity. This method analyzes RNA to determine which genes are actively being expressed at a given time. For example, In human gut, it can reveal which microbes are actively digesting certain nutrients after a meal. So, it tells us what microbes are actually doing at a given moment, not just what they are capable of. To go even further, scientists study actual products of microbial activity. Metaproteomics analyzes proteins produced by the microbes, the molecules that perform most cellular function. Metabolomics analyzes metabolites, the small molecules produced during metabolism. Together, these approaches reveal what microbes are actually producing and how they interact with their environment. For example, in fermented foods like yogurt, these methods can identify enzymes and metabolites responsible for flavor and texture. These approaches provide direct insights into microbial function and interactions. Each of these methods provides a different perspective, such as microscopy shows us where microbes are, DNA tells us who is there, RNA reveals what they are doing, and protein and metabolites show what they are producing. When combined, these approaches provide us a complete picture of microbial communities. Let's see the application of culture-independent methods. Culture-independent methods are not just powerful tools for research, they are widely used across many important fields like human health and microbiome, environmental studies, agriculture, biotechnology and industry, clinical diagnostics, food and fermentation, ecology and evolution. Let's explore some of their key applications. First, human health and the microbiome. These methods are extensively used to study human microbiome, especially the gut microbiome. For example, scientists can analyze stool samples to identify microbial imbalances associated with disease like obesity, diabetes, or inflammatory bowel disease. They also help us understand how microbes influence digestion, immunity, and even mental health. Next, environmental studies. Culture-independent methods allow us to study microbial communities in soil, oceans, or extreme environments. For example, in ocean ecosystems, they help us identify microbes involved in carbon cycling and climate regulation. In soil, they reveal microbes that play key roles in nutrient cycling, such as nitrogen fixation. Another important area is agriculture. These methods help analyze soil microbiomes and plant-associated microbes. For example, scientists can identify beneficial microbes that promote plant growth or protect crop from disease. This knowledge can be used to improve crop yield and sustainability. Now, biotechnology and industry. Culture-independent methods are used to discover new enzymes, bioactive compounds, and metabolic pathways. For example, novel enzymes identified from environmental DNA can be used in industries like biofuel production or pharmaceuticals. They are also important in clinical diagnostics. Traditional methods may fail to detect certain pathogens, especially those that are difficult to culture. Culture-independent techniques can directly detect microbial DNA or RNA from patient samples, enabling faster and more accurate diagnosis. Another key application is in food and fermentation. These methods are used to study microbial communities involved in food production. For example, in fermented foods like yogurt or cheese, they help identify microbes responsible for flavor, texture, and safety. Finally, ecology and evolutionary studies. Culture-independent methods allow scientists to explore microbial diversity across different environments and understand how microbial communities evolve over time. This helps us to understand the role of microbes in ecosystems and their response to environmental changes. Overall, culture-independent methods have transformed multiple fields by allowing us to study microbes in ways that were not possible before. To understand the strengths and limitations, let's take a deeper look at the comparison between culture-dependent and culture-independent methods. Starting with the core principle. Culture-dependent methods rely on isolating and growing microbes in artificial media. In contrast, culture-independent methods analyze biomolecules such as DNA, RNA, proteins, and metabolites. directly from environmental sample. Next, culturing requirement. Culture-dependent methods require successful growth of microbes in the lab, whereas culture-independent methods completely bypass this step. Now, looking at the microbial coverage, culture-dependent approaches capture only a small fraction, often around 1% of microbes. Culture-independent methods, however, detect the vast majority, including unculturable organisms. Now, the resolution. With culture-dependent methods, we can obtain pure isolates and study microbes at the strain level. Culture-independent methods typically analyze communities, but with deep sequencing, strain-level resolution is sometimes possible. For diversity detection, culture-dependent methods tend to underestimate microbial diversity. Culture-independent methods provide a more accurate and comprehensive view. Considering functional insight, culture-dependent methods allow direct experimental validation of microbial function. Culture-independent methods often predict function from genetic data, although activity can be assessed using RNA, protein, or metabolite analysis. In terms of quantification, culture-dependent methods use colony counts, which are semi-quantitative. Culture-independent methods usually provide relative abundance with absolute quantification possible using advanced techniques. Looking at throughput and scale, culture-dependent methods are limited in scale. Culture-independent methods are high throughput and can process many samples simultaneously. For time requirement, culturing can take days to weeks. Culture-independent methods can be faster once sequencing workflows are established. Now the cost. Culture-dependent methods are generally more affordable. Culture-independent methods are more expensive due to sequencing and computational needs. Regarding technical complexity, culture-dependent methods require basic microbiological skill. Culture-independent approaches require expertise in sequencing technologies and bioinformatics. Considering bias and limitations, both approaches also have bias. Culture-dependent methods suffer from culture bias due to selective growth condition. Culture-independent methods may introduce biases during DNA extraction, PCR amplification, or sequencing. For reproducibility, culture-dependent result can vary depending on growth condition. Culture-independent methods are more reproducible when standard protocols are used. When it comes to data type, culture-dependent methods provide distinct functional and phenotypic information. On the other hand, culture-independent methods provide genetic data, functional potential, and sometimes activity information. Finally, spatial information. Culture-dependent methods provide limited spatial context. Culture-independent approaches, especially techniques like FISH, can reveal spatial organization of microbes within a sample. Examples include plate culture, enrichment techniques for culture-dependent methods, and 16S sequencing, metagenomics, or metatranscriptomics for culture-independent methods. Overall, these two approaches are complementary and are often combined to achieve a more complete understanding of microbial communities. We once studied only the microbes we could grow in the lab. Today, with culture-independent methods, we can explore the vast invisible world of microbes that shape life on Earth. And this shift has transformed modern microbiology. Here, we have discussed culture-independent methods for studying microbial communities. We covered the main approaches, including microscopy-based methods like FISH, DNA-based methods such as 16S rRNA sequencing and shotgun metagenomics, and advanced techniques like metatranscriptomics, metaproteomics, and metabolomics. We highlighted how these methods allow scientists to study microbes without growing them, including the vast majority of unculturable organisms, and gain insights into their identity, function, and activity. We also explored their wide range of applications in human health, environmental studies, agriculture, biotechnology, and food microbiology. Finally, we compared culture-dependent and culture-independent methods to better understand their strengths and limitations of each approach. Overall, culture-independent methods have revolutionized microbiology by revealing the unseen majority of microbes and their roles in the ecosystem, health, and industry. That's all for today. Thank you, everyone.